8 is all about genetics. And this is one of my favorite modules, mainly because there's a lot of problems to work in it, and I really enjoy working problems because I really enjoy math. There's not a lot of math involved in this, but it is still a lot of problem solving. So hopefully you enjoy solving problems as well. You'll notice there's a lot more videos for this chapter just because I want to make sure that you're understanding how to really walk through these problems and how to solve them. So that's why you'll see a lot more videos in this module than you've seen in some of the earlier ones. We're going to start with example 8.2. I realize that he has done this on this companion CD, but I want to do it again on the board for you to show how it might be helpful for you to set it up on paper. One of the things that you have to be really careful about when you're working these problems is that you read the problem carefully and that you're making sure that you get the parents correct. So if you look at example 8.2 on page in your book, it says a heterozygous purple flowered pea plant is crossed with a heterozygous purple flowered pea plant. Well, heterozygous means that those alleles do not match. They're both different. You're going to have one dominant allele and one recessive allele, making it heterozygous. So a heterozygous purple flower is crossed with a heterozygous purple flower pea plant. So your two parents actually look identical to each other. You'll need to be extra careful when you're doing this because you do have capital P's and little p's, and they tend to look the same on paper, so you want to make sure that your handwriting is such that you can tell whether you've got a capital P or whether you've got a little p. So it's easier to use other letters sometimes, but if you do have something like p's, you need to make sure you know which is capital and which is lowercase. So because I have two alleles for each parent, it doesn't matter which parent goes on which side, and in this case they're identical anyway. So here I have a capital P and a little p, and here a capital P and a little p. So when I put these together, the top left corner is going to be two capital P's. The top right corner is going to be a capital P and a little p, as well as the lower left corner is going to be the same way. Bottom right corner is two little p's. So it's asking for genotypes and phenotypes, so we need to make sure that we do both. I'm going to start with genotypes. Okay, so I have three different genotypes. I have capital P's, and there's just one of them in my Punnett square, so that is 1 out of 4. I have capital P, little p, and that's in 2 of my squares, so that's 2 out of 4. And then I have little p, which is in just 1 of my squares, so that's 1 out of 4. In this particular kind of problem, when I only have 4 possibilities, it's not too difficult to make sure you have them all, but just as a point of practice, what you should do at this point is make sure that you didn't skip any. 1 plus 2 is 3, plus 1 more is 4. So I did have 4 total genotypes. Again, 4 isn't too difficult to make sure you didn't miss one, but we're going to be doing some larger Punnett squares later, where if you don't make sure you got all of them, you're really going to have a problem. So it's a good idea to add those up and make sure that you didn't miss any. Now, when you calculate these percentages, it's perfectly fine to use a calculator. This first one is simple enough that you may not need a calculator for it, but if you don't remember how to convert a fraction to a percentage, we'll run through it real quick. 1 divided by 4 will give you 0.25, and that is a decimal. To turn a decimal into a percent, you need to multiply by 100. So you're going to type into your calculator 1 divided by 4 times 100, and that should give you 25%. Okay? 2 out of 4 is half of them, and you may already know off the top of your head that that's 50%. If you don't, take your calculator, punch it in. 2 divided by 4 times 100, and that will give you 50%. And 1 out of 4 I already had from the top, so I don't need to punch it into the calculator again, so that's going to be 25%. And again, just to check yourself, go through and add those up and make sure that you actually come up with 100% when you get down to the bottom, because if you don't have 100%, you made a mistake someplace. Okay, so those are your phenotypes. Now we need to look at the phenotypes, which means what do each one of these look like? Well, if I have a capital P in the genotype, that means that there's an allele there for a purple colored flower. Purple flowers are dominant over white flowers, so both of these are going to make purple flowers. And so what that means is that 3 out of 4 of the offspring, the chances are that that will be purple colored flowers, and 3/4 is the same as 75%. 2 little p's means that I have white flowers, and I just have 1 out of 4, which I already know is 25%. And again, 75 and 25 makes 100%, so that I know that I have all of them. I want to do one more with you just to give you another example, show you how to work these problems, make sure you're really comfortable with this. So at the bottom of your notes, you'll notice that it says problem for video number 1, and that's the one we're going to go through. The allele for axial flowers, and axial flowers just means that it's on the stem. So a terminal flower is going to be up here, an axial flower is going to be down on the main part of the stem. That's what the terminology means. So the problem says the allele for axial flowers is dominant, it's a capital A, and I'm going to cross a plant that has axial flowers and is homozygous. Homozygous means both alleles are the same. If it has axial flowers, then it's going to be capital A, capital A. And it is crossed with a pea plant that is heterozygous. Heterozygous means the alleles are different, so I'm going to have a capital A, little a. Okay, when I run through this Punnett square, again, I have 2 alleles for each parent. Doesn't matter which parent goes where, you're going to end up with the same results. I'm going to put my 2 capital A's on the top. I'm going to put capital A, little a on the side. And then when I go through and fill my Punnett square, on the top row, I have all capital A's. On the bottom row, I have capital A, little a. The same thing is being asked in this problem. What are the possible genotypes, phenotypes, and percent chance of each? So we'll start with genotypes. I have 2 possibilities. I have 2 capital A's, and they're in 2 out of the 4 boxes. And I have capital A, little a, which is also in 2 of the 4 boxes. 2 out of 4 is 50% of each. And again, I double checked in my head, okay, 2 plus 2 is 4, I had all of them, 50 plus 50 is 100%. So if I didn't have 100%, I know I have a problem someplace. Now let's look at, whoops, not genotypes. Now we're gonna look at phenotypes, which means, what does this plant look like in real life? Well, with both of these, since they both have the dominant allele in them, both of these plants are going to have axial flowers on them. There won't be any with terminal flowers because I don't have any that have 2 little a's. So my axial flowers, I have 4 out of 4, and that's going to be 100% of the offspring of this cross are going to be axial flowers. Hopefully that provides a little format for you for when you're doing your problems as to how you can set it up on paper and make it nice and neat so you can see what you're doing with it.